on this webinar basically about developing an always active termite management system. Now, Joe DeMark is originally from Wisconsin in the United States of America. He's been a research scientist with Dow AgroSciences and Corteva AgriScience for more than 28 years. He graduated magna cum laude from the University of Wisconsin Whitewater with a BS degree in biology and a minor in chemistry. After earning his MS and PhD in entomology at the Purdue University in 1988 and in 1992 respectively, he joined Dow in 1992. Throughout his career, Joe has worked in urban pest management, research and development, and has had many projects, including general pest, termites, and fumigant products. He's been directly involved in the development and enhancement of the Sentricon termite colony elimination, and more recently, Sentricon AlwaysActive. He has had many roles over his years in urban pest management, research and development department, and his current position is field scientist and commercial zonal biology leader for urban pest management. He's located in Fayetteville, Arkansas. In addition to his field scientist role, he's also was global biology team leader for all urban pest management projects from 2011 to 2015. Joe has been invited as a speaker at numerous pest control associations and entomology meetings all around the United States. And he has also co-authored 16 refereed scientific journals, proceedings, book chapters, and several trade journal articles on urban pest insects. He is also author and co-author on several trade, he is also author, co-author on 21 patents, including the Recruit HD patent granted in 2015. He currently has an adjunct professor status at the University of Arkansas and previously held adjunct status at Purdue University. He was also the 2005 president of the Ohio Valley Entomological Association. So without further ado, firstly, I'd like to thank Joe for giving up his time. It is 7 o'clock in the evening over there, so we're very, very much appreciated and very excited to hear you share your insights over your fabulous career with Dow/Corteva. So just one housekeeping, if anyone does have any questions, we will use the Q&A function at the bottom of the screen. We will answer questions at the end of the webinar. And if we run out of time, we will endeavour to answer as many questions as we can via email after that. So without any further delay, Joe, I'll hand over to you. Thank you.
Okay, well thanks very much, Joe. I'm going to go ahead and share my screen here. Hopefully this will come up. Are the slides coming up? Okay. And okay, very good. So yeah, again, thank you very much. It's a pleasure to be here to be able to talk to everyone there in Australia. I had an opportunity to come to Australia back in 2012, and Joe George took me around Sydney and some other locations for a couple days, and I got to visit with some of the Sentricon customers there at the time. And I really appreciate this opportunity today. So I'm going to talk about the development of AlwaysActive, what went into it, with an emphasis on bait durability. So in our company, Corteva, previously Dow AgriSciences, we've had a real goal of continuous innovation in the Sentricon business. It's been a strategic objective of ours to set as a key success factor back as early as the early 1990s for the Sentricon system. So, you know, and it's kept me employed for 28 years as I've been doing work on Sentricon for 28 years. We have continued to sustain investment in R&D, and mainly this was to optimize our system performance over the years. If you've been using the product for years, you know that we've made many changes over the years. So we've tried to always optimize system performance, minimize component costs as best we could, reduce labor for servicing for our customers, and to build and build increased customer satisfaction. This included bait station designs, both above ground and in ground. We've gone through different termite monitoring devices over the years with the electronic sensing protection is not as critical anymore now that we have AlwaysActive. And of course, we've looked at, we have two different excellent active ingredients, hexafluorine and novafluorine, depending on what country you're in. And bait matrices, we've done a lot of improvements, and that's mainly what we're talking about today, AlwaysActive. So over the years, we've gone from many different iterations of our bait. We started with what looked like a sawdust bait called Recruit bait with wood flour. And we moved to what we call Recruit 2 and Recruit 3 over the years, where we had a laminated textured cellulose, which is kind of like looked like paper towel basically with hexafluorine sprayed on it. And then we went to Recruit 4 or Recruit with PTC, and that was the briquetted formulation. And then we moved to AlwaysActive. We call it in the US Recruit HD with AlwaysActive, or basically if I say Recruit HD, it's the same thing as AlwaysActive. And the HD originally stood for high density bait. The system itself, if you've been around for years using the system, we used to do a monitoring with wood monitors, baiting, and then back to monitoring. But with AlwaysActive, we were able to go from installation directly to annual servicing. So the Sentricon stations, as you know, are installed around a structure. And then we have the AlwaysActive or Recruit HD bait in every single station. And then all stations are checked manually, annually. And Recruit HD is normally replaced as needed. We say at least when a third consumed or more. I know maybe it's in some location we say at least half consumed, then replace it. So it's really novel bait technology when we developed this. It's a solid cellulose-based composition. It has the active ingredient of hexafluorine and novafluorine in some other countries, but hexafluorine at 0.5%, and then other ingredients that make up the polymeric cellulose-based formulation. So it's an extruded formulation with a polymer base with cellulose and or active ingredient. And it's formed in a novel manufacturing process and has many unique properties of the matrix. Again, high density for HD, for increased loading, much more density and much more bait than we were putting in stations previously, and I'll get into more detail of that with our previous formulations. We've found that it's very highly palatable to termites, very highly desirable. Termites really find it a very, very good formulation to eat. It's highly durable, and that's we're going to be really concentrating on the durability today, which is a key to being able to go out, once a year and get good control during that year. And again, the active ingredients immobilize in the dense, rigid polymeric substrate. So hexafluorine, and I think if you're familiar with the system, and you probably are over the years working with it, hexafluorine is in the chemical family of benzophenylurea. It's a benzoylurea, and the toxicity is expressed through the inhibition of chitin synthesis, and sometimes these hexafluorines note also as a chitin synthesis inhibitor. In other words, it doesn't allow the termites to molt. When they go through their molt, they get stuck in their exoskeleton and they don't get to the next stage. And I've got pictures at the bottom that we took in our laboratory of termites that were exposed to chitin synthesis inhibitor hexafluorine and get stuck and they don't make it out and they die at the molt. That's the main cause of death in the termites. Now, one thing about termites, and it depends on the species, but on average, it depends on the age of the termite itself and how many molts it's gone through, but on average, they molt about every 45 days. That's just the biology of the termite colony. And the younger termites might molt sooner than that, and older termites that have molted several times already might take longer than that, but on average, it's about 45 days. So it does take time, and it's just based on the biology. you know, the quickest you probably can get elimination is 2 months. Once feeding on the bait, it's like on average 2 to 4 months in the prime time of the year. And usually we get them within 6 months. But a lot of it's based on time of year, how much they're molting. And as I say, they molt on average about every 45 days. But that can change during the time of year also. In the spring, summer, they molt. The termites are molting more often, and in the fall, winter, they slow down and they don't molt as much. They don't feed as much either. So you guys are coming out of your winter season into spring, and if you get them feeding on bait now, you're going to get good molting and good eliminations coming up soon. Again, in the wintertime, it can take a little bit longer because they're just not molting as much. Some of the secondary effects with hexafluorine ovicidal properties, if the queen is still alive and she's laying eggs, those eggs, if exposed to hexafluorine, will not hatch because the young inside the eggs will die before they can hatch because they're not molting into the next stage within the egg to get out of the egg and hatch. There's also some other effects of chitin inhibitors and hexafluorine, and that the gut and the tracheal system of insects is also lined with chitin, and that, you know, we feel that that's another effect. So just a comparison, looking at trying to move this, the size or the baits themselves. The previous Sentricon bait, which was in the U.S., was called Recruit 4, a different type of Recruit bait, a hexafluorine, no-fluorine, was 65 grams. And when we went to the Recruit HD or Always Active, it went to 150 grams, so about 2.3 times the amount of bait being put out there once we went to Always Active. Trying to get this slide to go to the next one. Okay, so we did a lot of studies to develop Recruit HD over the years, or to develop Always Active bait, and we did a lot of these. And in the interest of time, I'll just kind of give the quick results on these, and I'll go into much more detail on the durability. But we did lab characterization of consumption and efficacy of all of key termite species that do molt, the lower order termites, and we found that Always Active was highly palatable and efficacious to the key termite species, and even as efficacious as our previous formulation. So we felt good about that. As far as hit rate and palatability, we did studies in the field. We compared Always Active to wood monitors, and termites infested and consumed Recruit HD much greater than wood monitors when we did them side by side in the field. We also did a study in the US. We had to get through this. It was a termiticide scientific review panel. It was scientists from government and universities that approved our protocol and set the parameters for what we needed to do in order to get Recruit HD or Always Active registered. And we did 54 curative trials, or trials that were on structures that had active infestations, and 59 preventative trials, structures not necessarily that had termites in them at the time, but we found termites on the property. We also did placebo or control trials where we just put out wood monitors to compare to our beta trials, which was a requirement of the trials that we did. Result was, of these 113 colonies, the parameters they set that we had to eliminate every single termite colony that we identified around those structures, the curative and preventative sites, within 1 year for 85% of those sites. And we did at 1 year 90.3%. And shortly thereafter, we got all 113 sites cleared. But we passed the 85% 1 year point. And of those 113 sites, we actually eliminated 245 colonies around those sites. And we determined colonies by doing the DNA. So we had good results on efficacy. And then we also wanted to look at if a certain, because we got 245 colonies, if one colony is feeding on one species, a different species, and a different colony feeding on a termite Always Active bait tube, will a new colony moving in feed on that, or will they not feed on it? And we found that new innovative colonies will readily feed on previously consumed always active baits. And in fact, sometimes they feed a lot more, and we're not sure why, but that's what we found in our research. The durability is what I'm going to go into right now, because that was the big key of the development of always active bait, was our durability, the ability to have a durable bait. We were always asked, why weren't you doing, you know, why didn't you just put bait out from day one? Well, our bait, when the previous Sentricon baits formulations would not last more than a month or two in the ground. So we had to go out there at least quarterly. And we just couldn't put out the bait from day one. We had to use wood and then go to bait. But with always active, we were able to go directly into the ground with the bait. And it's because of the durability that we're able to do that. We did some initial studies in the lab where we exposed always active bait to brown rot and white rot up to a year. And then we fed them termites in the lab where we exposed the fungal exposed baits, both white and brown rot fungi, to termites. And we looked at consumption and acceptance. And we found that there was equal or more consumption of the exposed baits versus non-exposed or fresh baits, and no differences in mortality for the ones that were exposed. So that was in the laboratory, but then we wanted to take this to the field. And that's what I'm going to concentrate on now. We've got over 10 years of data from baits in the field, from field locations, and we also are taking baits from commercial customer sites at homeowner sites and having them sent back in after 5, 6, 7 years. And then we're looking at those in the lab. And we're also putting them back out in the field, looking at termite acceptance and efficacy. And that's what I'm going to discuss here for the bulk of the rest of this presentation. So, active durability. So that, as I mentioned, I'm going to talk about this next. I'm going to give an overview of some lab and field trials that we did with Always Active. And these were actually given at a scientific meeting a couple of years ago. And I'm going to just present the same data that we presented at a national meeting in 2018. So almost 2 years ago, was at the end of 2018 when we did this, but I wanted to share these results with you today. So the first presentation we did at the national meeting was the laboratory performance of field age recruit HD or always active bait from US commercial sites. So, and some of these are from commercial sites, but I'm also going to share a little bit of data that we did from just some field aging sites. So we've actually, when we first started developing always active bait, this was way back in 2007, before we had a product on the market in 2010, 2011. We actually had the foresight to start putting out some of our always active baits into some field locations and let them age in the field, because we wanted to get an idea of how they're going to age in the field, and we wanted to get them out there early on. Our goal was to let them, we were hoping that they would last up to 4 years. And what you'll see today is they last, in many cases, up over 10 years in the field once they're installed. We also collected baits across a larger geographical area, with collaborating with what we call in the United States, and I'm not sure if it's a different term in Australia, but we call them certified Sentricon specialists. And this is our customers that use the bait, or the pest control management professionals, the pest management professionals in the US. The ones that use Sentricon are called certified Sentricon specialists. And we collected baits directly. We asked for their cooperation. We asked them to send in baits directly from their commercial sites that were aging in the field at all different locations around the US. Sent them back to us, and we looked at them in the lab for acceptance and what they actually look like. And then we took them back out to the field, and I'll present that information here. So before I talk about those commercial study sites, let me go back. We did have these 10-year study locations that I mentioned that we started in 2007, and they were in Fowler, Indiana. This is United States. Indiana is in the north central part of the U.S. We had a site in Wayside, Mississippi, and also a site in Clemson, South Carolina. South Carolina and Mississippi more in the southern part of the U.S., a little bit more hot, humid weather, and then Indiana was a little bit colder in the wintertime. So we had these 3 locations, and we just put out a bunch of bait in plots, and these are not around structures, just out in the field. We wanted to get a feel for how it aged. Starting in 2007, we did this, and we put the AlwaysActive Recruit HD treatments out, and we wanted to put them out in locations that were both irrigated, so we wanted to keep them wet, and then some of our locations were in natural rainfall. For instance, in Mississippi, we had one irrigated site where we irrigated and kept it wet, and the other site where we had a plot of stations with our baits out was more natural rainfall, just kept it natural. We collected baits from the field yearly, and in fact, we started quarterly because we thought we was going to just look at this for 4 years, but as I mentioned, we ended up going for up to 10 years, but once we realized they were lasting longer, we started taking less from the field, and we only started taking them annually. And I'll present 10-year data today because we have a few more out, but we ran out of baits at a couple of locations after about 10 years. But I can show you 10-year data, and we're going to continue doing the commercial sites so we get more than 10-year data in the future. But what we do with these baits is we bring them back to the lab and do a consumption assay looking at fresh versus aged material, these aged baits, and then we look at efficacy over 42 days to see if they're still killing termites after being in the field for up to 10 years. So the lab trial is pretty simple. We have a couple Petri dishes connected with Tycon tubing. We put 100 termites. We use Reticulotermes flavipes, which is a species in the U.S. that's the key main species that's mainly found in most all states that have termites in the U.S. And then we put an age recruit or a fresh recruit or AlwaysActive bait in the foraging chamber. We look at consumption over 7 days, and then we look at efficacy over 42 days. And the results of the 10-year data, and this is consumption. We have, just to give you an idea of what we're looking at here, we've got the blank control is the lighter brown on the left. We have fresh Recruit HD or fresh AlwaysActive is the yellow, and then we have the different age materials out to 10 years. Indiana, Mississippi, and South Carolina in the blue, the kind of orange and the green color on the right. And just to point out here, what you're seeing is the consumption of the age material, Indiana, Mississippi, and South Carolina, was no different than our blank control. It was actually more than our fresh Recruit HD in the laboratory. So, in fact, we've always kind of said that, don't replace the bait if they get a little moldy, if they get a little aged, because they actually tend to like it even more than the fresh. But once you're putting fresh bait out in the field, it starts, molding up really quick. So we feel really good. 10 years out, we're getting good, still good feeding on the baits. And then if you look at efficacy of the baits, this is over 42 days. We usually run an efficacy trial about 6 weeks, 6 to 8 weeks. After that, we lose controls, but we had a blank control of AlwaysActive blank bait. And we compared it to fresh AlwaysActive or Recruit HD, and then the 10-year age baits. Just to point out here, the fresh material is no different from any of the age materials out in the field for 10 years. And the number or the blank control was significantly more survivorship, which you would expect, no active ingredient, but the age material was no different than the fresh material. So really, really great results for us out to 10 years. And we've consistently saw this from 5 years on. We had these kind of results every single year. So now getting into the commercial sites in 2017, and we're continuing to do this every year. I'll present data from 2018. We collected them in 2017, and we collected sites. These are commercial sites, and the numbers in each one of these states are where we collected from our customers or certified Sentricon specialist sites. So we had like 20 sites, and this is Louisiana. We had 15 in Hawaii, And you can see the numbers here that were sent in. A total of 167 sites were sent in, and we asked them to send in 10 always active baits from their sites. And these sites were from 2017 that we asked them to send in from. And they were originally put out in 2010, 2011. So they were about 5.5 to 6 years old. And just to give you an idea, this is kind of a lot of information here on one slide, a lot of pictures, but we rated them in the lab. And this is just a representation of what we're calling low visible degradation of. These are individual sites, and you can see, yeah, they've got cracks and they're kind of, some are a little moldy looking and some are broken, but not too bad. But 50% of these total, and these are about 5.5 to 6 years out at commercial locations around the country, around the US, 50% had low visible degradation that we could see. And this again is a representation. Then we had a 34% were more moderate. You see a few more breaks, some bigger cracks here on these. Some are broken in half. Just to give you an idea, and this was about 34%. Again, these are almost out in the field for about 6 years. And then we had 14% had high visible degradation. You can see these are busting a little bit more, and especially the ones toward the bottom pictures are breaking apart. Some of this could have been some termite consumption, but a lot of it's just some degradation that we saw. Again, these are just randomly chosen from sites from around the US about 6 years out. And just to give you an idea of what we're calling high visible degradation, but of all those 167 locations that sent in 10 baits, we had about 14% that were highly degraded. Just a close-up. This one, there's an example of one from Louisiana in the United States, kind of near the city of New Orleans, which is in the southern part of the U.S., kind of near the Gulf Coast, near the ocean, and it's very hot and humid in this area. This one was out for about, can I move this bar here? It was installed May of 2011, so it's almost 6 years, about 5.83 years in the ground. And so that's kind of what that one looked like as an example. I show this because I want to show the cross sections in the next slide. Okay. It says my internet connection is a little unstable right now. I'm hoping I can go to the next slide here in a second. You can hang up a little bit. Sorry about that. We're having a little bit of delay for me to get my slide to move. Anyway, just as we're looking at this again, not a very, almost 6 years, southern U.S., not really bad as far as the degradation on this example. Okay, finally got to the next slide. Sorry about that. I wanted to show this same site where we took, and this is what we do in the lab, we take cross sections to kind of give you an idea what these baits look like. So if you look to the right, you can see some of the cross sections, 20A and 20B to the right. And what you're seeing is kind of a degraded edge, and you can see the crack. And in the center of the material itself has really good cellulose still remaining. And this is kind of interesting because as we looked at this in the laboratory, we looked at feeding, and a lot of times they'll feed on the inside and they'll leave some of the crust on the outside alone, but they're still getting a lot of the bait even after 6 years out in the field or 10 years. So here's the data that we looked at. This is consumption data after 7 days in the lab. And this is just examples on the right. I've got blank extruded or blank material, which is always active, is the yellow bar on the left. New material, new always active material is the second, the blue bar, and then all the green bars are from age sites as an example. And just to look at the consumption here, if you compare it to the blank or the new, a lot of these green bars are actually higher than blank material and the new material. A couple of them are lower, but there's a lot of variability. But overall, there were no significant differences between the new material and the aged material from these commercial sites. This is an example, and we saw this across all the sites. And then as far as survivorship, we're looking at number alive here, termites. And this was run for 5 weeks. We cut this one a little short. You usually have to go 6 to 8 weeks to get 100% control in the laboratory. But if you look at the control, which is the blank extruded HD, the blank extruded, the yellow on the left, every one of these, including the Recruit HD or the AlwaysActive new, and all the aged were at least as efficacious as the blank or as the new material. All the aged were as efficacious as the AlwaysActive new material, and all were had more efficacy than the control, which you would expect. Again, no active ingredient. What I want to show here, I'm going to show a couple graphs just to show a correlation here. This is the weight of the bait. And remember, these baits, they start out at 150 grams. And this is the weight of the bait by years in the ground. And you'll see most of these are around the 6 year. You'll see a lot of dots around 6 years here. And you'll see the overall average line goes down over time, but you know, the weight is still up around on average over 100 grams, 110 grams or so, even at 7, 8 years. We had a couple even older samples send in that we included in this data set, but most of them in the 6 year. But there's really, you know, there's some that are completely fine at 6 years, 150 grams and more, and some did drop off with pretty high degradation. So it's really no rhyme or reason. It's interesting to see that kind of data. So I'm waiting for the next slide to come. So again, not a big, no rhyme or reason why, but we do see us over time, we'll do see that the years in the ground, the weight will drop off some. Okay, I've got a little bit of slow process of going through my next slide here. Apologize for the delay. Okay. This particular slide I'm going to show here is percent degradation versus ambient temperature, the yearly average temperature. So on the lower part of the graph, you can see the temperature from, this is Fahrenheit, I believe, 45 degrees to 80 degrees Fahrenheit on average. And you see that the baits themselves, the degradation averages jump all over the place. And what this is kind of showing, there's no rhyme or reason based on temperature why these are degrading. So kind of interesting that we didn't see a correlation, and they kind of held their degradation average across these different temperatures. And again, these are samples that are pretty much about 6 years old from the field. And coming up on one more slide here, another delay. Again, I apologize for delay in slide movement. The last one I'm going to show is based on rainfall. And we'll get to it here in a second, but the rainfall, I can tell you, same kind of data. We didn't see a correlation. You'd think the higher areas that had higher amounts of moisture, higher amounts of rainfall are going to show more degradation, but that's not what we saw, which was very interesting. We did not see a big change in the rainfall amount. I mean, as far as degradation goes, the higher the rainfall, in fact, did not show higher degradation. Wow, really having trouble getting this slide to move. Hopefully we'll get it to move here in a second. Okay, finally. So this is the one that's the rainfall one. And if you look at this, average annual rainfall from 0 to up to over 100 inches of rain. And if you look at it, in fact, if you go up higher on annual rainfall, the degradation, percent degradation even drops some at the higher. And again, no real correlation here on what's going on, why. But overall, you do see actually a lower amount of degradation at some of the higher, as the rainfall actually got higher. Again, having a slight delay here. Oops. Yeah, I apologize. Makes it tough sometimes when you're in these types of presentations. You're kind of relying on the technology, and it sometimes slows down on you. Okay, so the next thing I want to talk about is when we took this to the field, the hit rate and efficacy of these field age termite baits when redeployed in termite active areas. And again, this was another presentation we gave at this national meeting of the Entomological Society in the US. Wow. Okay, so the background is Recruit HD, of course, our bait was launched in 2010, 2011 in the US. We asked, again, I talked about this a little bit already. We asked our certified specialists to send in the baits. We did a lab analysis, and then for field and then also we did a field redeployment, and I'll talk about the field redeployment in this presentation. Again, same 167 sites that we got the bait from is what we redeployed in the field. We had a key question: our age Recruit HD always active bait showing degradation of the outer surface, which I kind of showed in that cross section. Are they attacked and fed on as frequently as the new Recruit HD baits or new always active baits when installed around infested? We had termites in infested trees. We had them in billets. Billets are kind of like a big tree trunk that cut from a tree and put out in the field and allowed termites to infest that, and then we put stations around that. There's also bucket stations that we have wood in, and we infest, we'll put bait around that. And also in one location, we actually just had a hit Sentricon station and baited around it. And these are randomly selected. The baits came from our lab. They were just randomly selected, shipped out to different people in our field, like myself, doing research, and then we put them in around these billets, trees, and bucket traps and or stations. So to do the hit rate trial, we put stations 1 foot from a hot station, billet, bucket, or tree, at least 1 foot and 1 foot away from each other. We had 3 treatments. We had the age treatments from the commercial sites, randomly selected. We also had a new Recruit HD always active bait and a wood monitoring device, which was our Sentricon with ESP device. We collected data on hits, yes or no, number of termites, termite species, percent consumption, and we evaluated monthly for up to 6 months. I'll probably show you data here, I believe 3 and 6 months for brevity. And then we looked at statistics on hit stations and then consumption of the baits themselves. And this is kind of the setup. It'd be a hit station, bucket, or a billet. And 1 foot out, we had 3 treatments: an age, a new recruit, an age always active, a new always active, or an ESP wood monitor. So we did the hit rate trial, and we did locations in different locations in the US, New Orleans, southern US, and we looked at different species of termites in the US. Coptotermes formosanus, the Formosan termite. We looked at Reticulotermes flavipes and some other Reticulotermes species, variations of Reticulotermes species in Mississippi and Florida. as well as Delaware, which is on the east coast of the United States, kind of the northeast coast, and also out western US in the desert, the Arizona desert termite, heterotermy species. So looking at the percent hit, again, these are aged versus new versus ESP. And just want to point out our hit rates, and this is after about 3 months of being in the field. We're looking at the age being the darker red or orange color versus the lighter tan compared to the new. You can see that the age are hit comparable, and in no case are they significantly different from the aged, and in most cases definitely hit higher than the ESP would. At 6 months, we saw the same type of data, just a little bit higher hit rates now, but 6 months. So no effect on hit rate on the age versus new. And again, in fact, in some cases, we actually saw higher hit rates on aged. As far as percent consumption, again, at 3 months, Louisiana here on the right, the LA, Louisiana, you see the higher percent consumption at 3 months. That's a Formosan termite. It eats a lot. We actually had a significantly more consumption on the AlwaysActive aged and new AlwaysActive versus the wood. All the other locations, we did not have a significant difference between aged or new or the ESP wood. Some of these Florida sites, Florida 1, Florida 2, the species that we used there was kind of a weak species called Reticulatermes nelsoni, a newly found species in the area, and actually was in a pine forest. And I didn't really feel that it was kind of a wimpy termite, didn't consume a lot. And I didn't feel like it was similar to what we would see around structures. That's why the numbers are lower here. And then at 6 months, similar type of data as we saw at 3 months. And as well as this is data just for the hit stations only. What I showed previously was all the stations hit or not. But when you're looking at just hit stations only, again, you're seeing really high hit rates for the aged and very comparable and not significantly different from the new, and usually more consumption for the most part versus the wood monitors. And here's some pictures I wanted to show. So I'll show you that cross section of the bait itself. And as I mentioned, as these baits degrade in the field, they get kind of a crust on the outside. And what we found is the termites will go into the center and eat the center out first. Eventually, these are Formosan termites in New Orleans. Eventually they'll eat the crust, but they kind of save it for last. This one on the left, you can see the nice AlwaysActive in the center that was still there and the crust on the outside. And then you can see one that they went in the center, ate the center out here in the lower bottom picture, lower right. And then they kind of left the crust behind. Same type of thing we saw with Reticulatermes flavipes. Here's a look down into the picture on the left is down into a bait station. It's the crust on the outside, but the bait on the inside that had the good cellulose itself was completely eaten out for the most part, except for this little plug that was left in there. Thought these were interesting pictures. When we first saw this, we thought this was pretty interesting in the field. What I'm seeing sometimes is just little pinholes sometimes in these. And you almost have to break them open to see, actually they're eating the inside out. And you don't really realize it sometimes until you actually break one of these bait tubes, those AlwaysActive tubes. This is from Arizona. This is a desert termite. You can kind of see little holes in the one on the left, the age. They pop into these baits, and they kind of eat from the outside in. There is some tunneling on the outside. Whereas the new, they kind of eat it from the outside in, the newer bait or the newer AlwaysActive versus the age. Just a different feeding pattern, but the results are the same as far as efficacy-wise. So we did look at the same baits for efficacy, and we did a colony elimination trial. So we asked, do the age baits show degradation of the outer surface, eliminate termite colonies as frequency as the new AlwaysActive baits when installed around infested trees? And I'm going to show you some data here. Around infested trees, billets, bucket stations, or active stations, and the answer is yes, and I'll show you the data here in a second. So in this case, we put a minimum of 6 Sentricon stations with active bait around an active billet an active station, an active bucket trap, or an active tree. Sometimes the trees, we only had room for 4 to 6 baits, but we just baited them around these active stations. So we know the termites are there, and then we baited them, and then we looked at the tree or bucket station or station if the termites disappeared after feeding on the bait for elimination. And that's how we calculated days to elimination. So we looked at aged recruit HD bait and new recruit HD bait. We collected numbers, data on numbers of termites, the species, percent consumption, and then the sites. We looked at 10 different trees, so 2 different treatments. That's 20 trees, 10 reps, 8 reps of buckets in Delaware and 9 reps of Florida, so you can double the numbers of buckets or trees on these. And what we found, a simple find here, was that the age baits were eliminated, just as well as the new baits. In fact, no significant differences at any one of these locations. And actually notice here that all of them were eliminated for both the aged Always Active and the new Always Active within 90 days. So within 3 months or less, they were eliminated. So very good results across the board for this, for the age versus our new Always Active baits. So the conclusions, there were a few differences in consumption and hit rate in our age versus fresh baits, with possibly a slight preference for the aged in some cases, but not always, and usually never significantly different between the two. For colonies that were eliminated, there were no differences in days to elimination, at least statistically, or in the number of baits consumed for the field age versus the fresh baits. And in the US right now, when our customers go out to replace the bait, that look to replace bait or check the sites, we tell them to replace our baits when they're broken, if they're like cracked in half or broken or degrading to the point where they're actually broken, so those highly degraded baits, or if they're more than 1/3 or more depleted by termite consumption. That's when we tell them to replace. If they're still, intact baits that are aged, termites like them, we tell them not to replace them. There's no need to replace them unless they're broken or greater than 1/3 depleted by termite consumption. And this is important for us to tell our customers that. There's really no need to replace, because even, in fact, the age baits, sometimes they actually like them better. They hit them a little bit harder and like them better. So we are continuing to look at this. If we do find a breaking point, we will let everybody know that, hey, here's an example of how you should be replacing these, but as of right now, that's our advice when broken or more than 1/3 depleted. I only have 10 minutes left. Again, I apologize for the slow advancement of some of the slides. It's technology, I guess. I did want to mention a couple things on above ground baiting, because I really think it's important to use the tool of above ground baits. And I know you've got Sentricon above ground bait in Australia. And really, it's one of the few baits that are out there on the market that you can actually use to do above ground baiting. In the US, it is the only bait, and it's the only tool that can be used in a structure to bait a colony. And the Sentricon AGs and the AGs used in the US, wonderful tool. They're a great tool. I really recommend using them. You use them on any termite infestation or swarm site in a structure. They can be used. It's better to put them on active infestation. Swarm sites, if you catch them right away, they're good. If they're swarming from a location and have swarmed after a few days, sometimes the termites leave and you don't get a good hit rate. But if you can find active termites, I highly recommend getting these up, because this will just get you immediate feeding into the colony. You don't have to wait for them to come in to find in-ground stations. They're placed directly on activity, and they really can address your property owner's immediate concerns. It can really help with the sale and having them understand what you're doing. So there's 100 grams of the PTC matrix in these stations. It's really a low impact application, direct baiting for immediate consumption, successful placement. You don't get 100% hits on these, but you really only need one to eliminate a colony. is what we found. If you get one hit and they eat most of that bait, we're eliminating colonies, even before they find an in-ground station sometimes. No other liquid or bait has this unique targeted approach. It can be used with Sentricon in ground, and we recommend that in the US. But it can also be used as a standalone. And all the research we did to look at Sentricon AG, and I know it was the same way in Australia, because when I was there 8 years ago, I worked with our scientist there, who has since retired, Paul Downard. That's exactly how he was doing the protocols. I worked with him on those, and that's exactly how the protocols were run. Just a few things about, and I think if you're using these, they're very simple to use. Want to inspect the site, which is Sentricon AG will be attached. If you see signs of activity, especially mud tubes, attach the housing, line the back. Don't break, break the tunnel in the middle, but don't break it on the edges. Let the termites feed through the tunnel into the bait when you install it, and use the breakout points in the hard cover to allow for attachment. Prepare the bait matrix, or open one end of the flexible bag in the station. Add sufficient water to thoroughly moisten the matrix. Knead it to like a dough-like consistency. Expose the matrix by cutting an opening for the entry of termites, where you put it on the wall or on the side of a structure. You securely attach the cover with appropriate materials. We use a lot of latex-based caulk. We don't like silicone-based caulk as much, because we found that it's high, you get some of the volatiles from that will temporarily repel termites, but after it dries, they don't. But we just tell people not to use silicone-based, but tape, non-volatile adhesives, and other appropriate materials. And this really is key. Getting a good seal around your AG, Sentricon AG, is really critical in getting an active hit in these stations. If it dries out fairly quickly, the termites will disappear. But if it's sealed really well and you keep the moisture in, you'll get good hit rates on these and get good results. And just one little bit of data here. We did a couple trials a few years ago. I show this, we did a lot of trials actually, but here are a couple different species. What we're seeing at the good time of year, which is spring into summer, we're getting eliminations, and months elimination of the blue bars, and these are just some different site names at the bottom. The mean average was less than 3 months to get elimination, and this is above ground baiting only. We did see high consumption in these, up to 600 grams. That's a total of six above ground stations being consumed, but it varies. But if you can get one consumed, you're in pretty good shape. The Tampa site, we had just barely one, a little over one bait, above ground bait consumed, and we had elimination. So average elimination time is just under 3 months, and these are great. And I just wanted to put a plug in for these. And in the US, we have some customers that use them all the time, and then other ones we can't talk them into using them. But once they start using them, they don't go back. They use them all the time. So I just wanted to point that out. And with that, thank you very much. I believe there is a little bit of time for questions, and I can hang around as long as need be.
Lovely. Thank you so much, Joe. As I said, we do appreciate your insight, an excellent insight into what is involved in the development of the Sentricon AlwaysActive system, and really how its durability and palatability is market leading. And it is also very interesting to see that what research just continues way after a product has been brought to the market. It just doesn't stop, does it?
Yes, it's something that we're continuing to do. I've been in the Sentricon research my entire career, and I'm hoping to retire still doing Sentricon research in a few years. And I hope I can just keep it going. And I think the company is supporting it, still supporting Sentricon. We're getting resources every year to continue research and get more answers and improve the system yearly if we can. Yeah, no, that's great to hear. All right, well, we do have a few questions, Joe, so I'll pose the first one to you. In many grass, the number of termites alive after 5 weeks, not consuming active ingredient, is around 70 to 80%.
How do you account for the loss of population when not eating active ingredient? Is it age?
Well, no. So you're talking about the lab trials. Yeah, the lab, it's really hard to keep termites alive in the lab. And be honest with you, if you can get 70%, 80% survivorship in controls, you're doing pretty good. It's not a normal field type environment. And, if you can keep the controls alive to that 70, 80%, you're doing well. So what we're trying to do is get at least 70 to 80%. And then if we can see that, we can usually get a good comparison to the ones with the active ingredients. So it's just based on the ability to do the lab work, and you're going to lose some regardless. It's not based on the active ingredients, it's based on the lab environment and keeping them alive. It's a lot of methods have gone into improving that, and 70 to 80% is about as good as we normally see, believe it or not. So.
Okay, lovely. Okay, the next question is, are you able to compare United States termite species used against the consumption, etc., of Australian termites, such as copter termes and Shedderina termes?
Yes, I think, in fact, luckily I had an opportunity to go to Australia myself, and the Coptotermes species in Australia, Coptotermes aciniformis, I got to witness the feeding and the how active and aggressive those termites are, and I do a lot of work on Formosan, Coptotermes formosanus termites here in the U.S. I did not see a difference, really. They're just as aggressive in Australia. And I think the results going out with Paul Downard years ago were very similar for Coptotermes. Now, Shedderina termes is a species we don't really have in the U.S. But my own experience with that species when I was in Australia, and I had experience in some other locations, it's very similar to what I believe is our Reticulatermes flavipes species, which is our most common species in the U.S. And I think it's similar to that as far as aggressiveness and bait consumption and colony elimination. So I think there's a pretty close comparison. Again, they're different species, but I think we can compare our native U.S. species to Shedderina termes, and the Coptotermes species in the U.S. compares, I think, fairly favorable with the aciniformis Coptotermes species in Australia.
Lovely. Okay, another question. How far away or from a property can a pod be installed?
Could you repeat that last part of that question again?
Yeah, sure. How far away from a property can a pod be installed? A station, I guess.
Oh, okay, I got you. Can a station be installed from a structure? Well, we recommend, we like to keep it, of course, some of these structures were treated with a liquid termiticide years ago, and that's the same case in the U.S. And I'm sure we try to at least keep a few feet or at least a meter or so out from the structure. But I think you can go out, 3-4 meters without being too concerned. I wouldn't want to go out much further than 2 to 3 meters on average. It just, you might run into more colonies. We do know, especially with the Coptotermes species, that the colonies can be huge and cover a lot of ground. So if you are a little ways away from the structure, say a few meters to 2 to 3 meters, I think you'll be okay. I would say if you can get as close as you can within a meter or two, if it allows it, just because you want to make sure you're getting the colony that's affecting the structure, but you can be further out, especially with Coptotermes species, because their colonies cover so much ground.
And I'd just like to add to that, Joe, is that here in Australia, our label requirement is 300 to 500 mil out from the foundation wall, but not in a chemically treated zone. Okay. Yeah. And I'd also actually just like to reinforce that our monitoring intervals here in Australia are 3 to 6 monthly, not annual inspections.
Okay, so you're at 3 to 6 months. Okay, we started with 3 months on the AlwaysActive, and we were able to show our data to the U.S. Environmental Protection Agency, and they were okay with us going out to a year. Yeah, I mean, maybe that's something down the road. We'd have to do more research sites probably to get that approved in Australia, but
and I think it's a difference between noviflumiron and hexaflumiron as well.
It could be too. I'll be honest with you, they're both very good active ingredients, but that could be the difference. They both work extremely well. If you're feeding on a bait tube and they're eating an entire bait tube of noviflumiron or hexaflumiron, you're going to get elimination of that colony. No doubt in my mind. As far as our customers in the US, we have an annual label. They can go out to a year. A lot of them don't feel comfortable doing that. They might go out every 6 months. Some go out still quarterly, every 3 months. It just depends on how comfortable they are. But by label, they can go out up to a year, but they can't go past a year within reason.
Okay. Yep. All right, we have another few questions. It is my understanding that the larger the station, the better the opportunity for interception. Is that, if that is the case, why is the Sentricon station small?
Yeah, that's a good point. I mean, that's a good question. Basically, you know, there are some stations that are a little bit larger. There's been some work done on that. I think the studies that were done on that were a little bit flawed. Unfortunately, the work, they looked at the stations every day. They pulled bait out or pulled wood out of the stations to look at hit rates. They actually used an older classical Sentricon with 2 wood monitoring devices with this really older design with the extractor up the center. And they looked at it every day versus another station that was larger that might have had wood in the bottom the whole time. But basically, we feel very comfortable with our design being out a product for over 25 years now, that based on termite biology and behavior, they're continuously foraging, and they're going to forage. They forage in a tree branch type pattern. And if we put them about 3 meters apart around a structure, regardless of the size of that station, we feel very confident based on our track record of success that termites will find these stations, regardless if it's, you know, so many millimeters wide versus another one. They're going to find them. They just, based on their foraging behavior and biology, they find our stations. And I'm not concerned about the size of the station. And again, that's based on experience and 25 years of doing this kind of research. They find them. If the termites are there, they find them.
And I think particularly from my experience with the trials that we've done here in Australia and what pest managers tell me on a weekly basis is that certainly with the introduction of the AlwaysActive termiticide rod in every station, due to their palatability, termites just, they'll preferentially seek them out as a quality food source.
Well, yeah, if they find them, they find it highly palatable, highly desirable, and they hit it. Now, I've done this work over the years. We had different kinds of wood in stations. I know you guys use eucalyptus in Australia, I believe, or you did before AlwaysActive. We used like a pine wood, and I used to see termite tunnels on pine, and then they would leave. I don't see that with AlwaysActive. When they find AlwaysActive, they hit it, and they'll feed on it. When they used to find some of our wood monitors, sometimes they would feed on it, and other times you would see, oh, they found it, but they're not here because they left and maybe didn't find it quite as palatable. But that's a good point, Joe, that the palatability has really helped with that also, as far as the foraging biology. And then when they do find them, they stay there and they hit them. Yeah,
absolutely. And that's certainly what we did see with the trials. And, you know, even if they've consumed a full rod, because, and you place a new one in, we never saw abandonment, very rarely, because of the pheromones.
It's really rare. Yeah, exactly. And usually, I can tell you, when we first started doing this work, we did our research sites, that TSRP protocol I mentioned early on, where we did the 245 colonies, 113 active sites, structures. We did it quarterly. So we looked at them every 3 months. And initially, when we first started doing it, we would come out after the first quarter, and these baits were hit hard after 3 months. And a lot of times the termites were already gone. And we were like, are they abandoning? But then we would do the structural inspection, and we would check every station. We had wood monitors around the street, and they were gone. So not only do they hit them quickly, but we got those eliminations quicker than we had seen before. And it's based on the palatability of the bait and the fast hit rates we would get.
Absolutely. Another question. How far apart would you recommend on a brick veneer dwelling? I assuming they're meaning how far the stations should be placed apart, I would think.
Yeah, I still think about 3 meters apart. You know, what your label says is correct. Yeah, I mean, regardless of the structure type, once the colonies hit it, you're going to get elimination. You get consumption of the bait, you'll get elimination. So I don't think there's any reason to go any closer together than what your label would say, or what your recommendations are, your technical manual recommendations are. Yeah.
Absolutely, and it's 3 meters and always has been. So
3 meters, yeah. Yeah, that's exactly that's the same thing we'd say in the US. So.
Yeah. Next question. You mentioned Novafumeron. There is another company using Novafumeron active. Why has Sentricon stayed with hexafumeron? I think the question means that Novafumeron is being used in the US and I think Japan as well, isn't it, Joe? Japan.
It's in Japan. There's a few other countries using Novafumeron. It's based on regulatory and getting it registered in the different countries. And for whatever reason, Novafumeron wasn't registered in Australia. And I don't know if that's ever going to happen or not, but there are some regulatory reasons. Early on, Japan got in on Novafumeron, but some of the other countries did not. Some of the European countries, like France, use hexafluomeron. But I'm not sure if you're talking about another company. There is another product out there with a product called Novaluron. It's similar to Novafumeron in name, but not the same. And maybe that's what the question was, but that's generic active, and so anybody can use Novaluron, but we have the patent for Novafumeron until, I believe, 2032 with AlwaysActive. So, you know, you could possibly see it somewhere else, but our AlwaysActive bait matrix patent is good for another 12 years, I guess. So you probably won't see a bait matrix like ours, but you'll see other companies try to come in with different chitin-sensitive inhibitors, which is not surprising since there are generic ones out there.
Yeah. Alrighty. Next question. I understand how palatability is of utmost importance. It's a bait stick, not bounded with plastic in this case. I would think this may deter a termite. Your thoughts?
Could you repeat that last third of your question there about the plastic?
So basically, I understand how palatability is of utmost importance. If a bait stick, not Sentricon, is surrounded with plastic in this case, I would think that this may deter a termite. Your thoughts? So in other words, like if the rod was sitting totally enclosed in a plastic casing, would that deter the termites? But not Sentricon, any insect growth regulator.
Completely in a plastic case? Are you talking about our station or no? That's because it's got the openings.
Yeah, the actual bait itself, I think.
If the bait itself, you know, of course the bait is an extruded bait, the AlwaysActive. It's got a polymer in it. It's got a little plastic in it. I'm not sure if that's part of it. However, we haven't seen that deter termites at all because they like the palatability. If it was totally encased in plastic, obviously it would be hard for the termites to get to the bait. I'm not sure if I'm answering the question correctly or understanding the question, but As far as being encased in plastic, I would think it'd be very difficult for termites to find it.
Yeah, okay. Another question. Basically, from a technician perspective, if the issue of the rod being eaten from inside out, but it sort of looks fine, how would the technician know that it's empty on the inside, basically?
Yeah, and that's a good question, because we're, you know, this new age bait and redeploying them in the field, and we're learning about this. I guess, you know, and I just did a trial last week where I checked a trial because we're continuing to do this work. A couple things I noticed that could be helpful. If a technician, first of all, he pulls a bait out, you'll feel that it's lighter, for one thing. Look for pinholes that I saw some last week that were age baits, you know, that were redeployed in a hit rate trial that I'm doing right now. Pinholes actually had little termite heads popping out. But the other thing you can do is pull that little plastic top off the bait. A lot of times you just take it right off the top, the green top, I believe, on your bait. And if you can sometimes look down into it and see, and I did that last week. In fact, I had one with a couple pinholes. I pulled the top off and I could see that there were 100 termites or more inside feeding and hollowing out the bait. But that's a good question. Until you actually do it and get used to seeing it, you don't really know. And you might actually think that you might go to a research site that has some older baits at it, or just a customer site, a home structure site, not necessarily a research site, but go to that and you might think, oh, the termites aren't even hitting these. And actually, a lot of them might have termites in them. You're just not seeing it. So it is kind of a learning thing, especially as the baits get older. Look for pinholes, look for termite. You got to give it a few seconds and not just put it back in the hole or back in the station right away. Examine it well. And if you suspect something, pull that plastic top off and take a look. You'll be able to tell pretty quick if you do that.
And you probably could also use a cake skewer to test the integrity of the rod. Use it.
Yeah, you probably could do that too. Yeah.
If it's empty.
Yeah, I mean, if it's really super light, you might even, you know, see it. You know, you don't really want to break a bunch of them, but if it breaks pretty easily and you find the termites in there, obviously you're going to want to replace it. But
yeah.
But yeah, there's different ways. But again, it may be tough to see it right away, especially with the older baits, where they're eating the inside out.
Yeah. Okay. We have one question on the above ground stations. Some people, we know, obviously have the perception that, you know, above ground baiting is slow and takes time. You know, what are the benefits of above ground baiting versus dust and foam on an active infestation?
Yeah, so especially first of all, if you're dusting or foaming and you're not using an in-ground bait around the outside, you're basically just stopping them in that spot. It's a spot treatment. If you have our in-ground bait around the outside, we recommend not doing a spot treatment with a liquid, because what you're going to do is going to move termites from one part of the building to another. If you can get bait feeding directly immediately, yeah, it's going to take some time, but you're going to eliminate that colony rather than move it around. And the other thing we found that with liquids, especially if there are some customers in the US that are actually doing a liquid treatment around a structure and then putting Sentricon around the structure. And we don't recommend that either, because what we've seen in some of the newer research, in fact, is the termites will die in an area, and then the dead termites themselves will become deterrent to other termites. And then in other words, if you're treating with a liquid around a structure or in a structure, termites will die. They might actually stay away from that area to the point where you're not going to get hits on your in-ground stations to eliminate the colony. Really, even though it takes a couple months to get elimination, if you can get that bait in from day one with an AG, there's really no better way to do it as far as getting rid of that termite colony. And once they start feeding on the bait, they're feeding and the structure will slow down. You might not get elimination within a few months or so, but as the termites die, they're not affecting that structure anymore either.
Okay. All right, we've got one last question. I think we'll probably make this the last one, as we've been going quite a long time now. was actually, okay. If a big area around the house is covered by concrete, what is your advice in installing them in concrete?
Yeah, so, and I don't know if in the US we will do a lot of coring. And I don't know if you guys core first a hole and then put a station in them or not. But we do an awful lot of that in the US, especially in the southern US where you have Formosan termites. And we get very good hit rates. Now it's a lot of work, you know. You got to get out drill a core and core some holes out. And a lot of our customers will actually hire a company to do that for them, and then they'll go back in and bait. But we get really high hit rates in coring in the US, and I would recommend doing that if you can, as best as possible, and try to stick to that every 3 meters or so around a structure. If you can core, I go for it. And I don't know if you guys do that or not, but that's what we do in the US a lot. So
yeah, we do here as well, Joe. Yeah.
Okay.
And the installation guidelines is exactly the same as if they were going in the soil, but they're just going through a poured concrete hole. And then we use a steel core cap that lays flush with the concrete.
We do the exact same thing, yeah. Same, we try to stick to the same, you know, every three meters or so around a structure, and you use the metal top on it. What we've done in the US a lot is we use the classic Sentricon station and cut the wings off and stick those in the hole. Whereas, you know, and I don't know if that's what you guys do too, but that works pretty well. We get, again, our hit rates are high. And in fact, I've asked for some data from our southern parts of the US from our customers. And I actually got a set of data that we're going to look at coring versus non-coring. And try to maybe put either a technical document or a publication out on how good it is and how much it helps to do baiting with AlwaysActive and coring.
Yeah. Well, we do use the old style station, Joe, throughout all concrete holes, but typically they leave the wings on. They just core and
they do. So if it fits in there, yeah. Some of our guys will just cut them off, but yeah, if it fits in the hole that you core, yeah. And that's exactly what we do. So same thing.
Yeah.
Can I just add from a construction perspective that if the pest manager has discussed with the builder the termite, that they're going to use Sentricon for a termite management system, some people are actually putting in PVC pipe before the concrete is poured, so that those holes are already available. Make it nice and easy, so you're not coring through brand new concrete, which is a nice, neat system for the homeowner.
Yeah, absolutely.
That's a good point. And we have done that in the US too. In fact, New Orleans, they built an entire building like that where they put the PVC in while they were pouring the slab of the structure, and then they put Sentricon baits in the entire building all the way around it. It was built in just like that.
All right. Yep. We don't have any more questions, so I think it's probably the right time to finish off this webinar. So thank you everyone for attending. You've been very good, some good questions, and I'm sure you found this extremely valuable. And again, Joe, thank you so much. We really appreciate your time. It's nearly 8:30 and you're probably hungry, I don't know, but
oh, I'm good. And I really appreciate the opportunity to talk with you all in Australia. I really enjoyed my visit there. I wish I could come back again. They're not letting us travel too much, but really enjoyed my visit there. In a great country, and really a neat termite business down there also by you guys. So really appreciate the opportunity to talk to you today.
Thank you very much.
Okay.
Thank you.
All right. Thank you.
Bye-bye.
Bye.